Okay.
Yeah, that's fine.
Good morning. I'm Dr. Sania Sofra, and we're going to be talking about DNA and protein damage in skin disorders. And just to give you sort of a little bit of a introduction, this is a DNA strand, you all know, DNA. The RNA is what carries the information to the cytoplasm. This is a protein. This is how the proteins, you see that's a sensory protein, it comes above the cell to sense. The action proteins are more inside the cell. That's again sensory protein, and this is like ion channels that are also proteins. And proteins communicate. You see this is two cells, and the proteins inside the cells are sort of communicating. And this is how it would come. It's like a graphic, but this is how it would look under the microscope. And basically, we have the cell and the nucleus in the cell. From the chromosome, you have the histone, and the DNA helix happens with all the genes. And then the DNA polymerase, it's a protein that comes and separates the DNA into RNA, the messenger RNA. And if there's any problems, it fixes it as well. And the messenger RNA will go to the cytoplasm. And these are the amino acids, which are going to, through the transfer DNA, will start kind of forming the proteins, which is like some beads, like a necklace that is all twisted around, and that's the protein. What we need to, what I would like you to offer you is that in physics theory, there is a new theory, it's called string theory. And according to string theory, everything is formed by strings, resonance strings, that if they resonate one way, if they vibrate one way, they create a proton. If they vibrate a different way, they create an electron. So proton and electron are very different things, and yet the basis of them is exactly the one and the same. And basically, it's like an elegant universe, which is like a musical symphony of resonance strings forming elements that eventually form bodies like our bodies. But the particle is extremely tiny, so it's 10 to the minus 38. The atom is 10 to the minus 15. A protein is 10 to the minus 9. And as you're advancing to these different levels of complexity, you form a gestalt, and a gestalt is more than the sum of its parts. And so the resonance cells will form an organ, and that organ is going to form, basically, it's going to start working with other organs, again, through the resonance process. And all that is going to create a phase, which is obviously a much higher level of a gestalt. Now, there is a time reversal. Scientists are trying to invent time reversal for a very long time. And 2017, the TARDIS is a new way of kind of a mathematical formula that will reverse time. And according to Einstein, time could go both directions, could go forward and backwards. But the way we understand time, it's only going forward. We cannot go back in time. And the reason why is because a higher level of complexity, like a person, is a gestalt. That's more than the sum of the parts. And this is a gestalt, just you can see here the face, and the guy with a saxophone, and the birds turning into fish. And basically, it's different levels of complexity. And if we went back in time, it would be like a puzzle that scatters its pieces all over the place, and it's very, very difficult to put it back together. So time reversal is only possible in the smallest scale, and it has been achieved, but only for very, very tiny elements. Nothing that is more gestalt-like. So in order for us to think about anti-aging we have to focus on molecular mechanisms, on the tiny aspects of our bodies. And if we do that, you see things like the G proteins or the ATP, they go routinely back and forth in time by turning into ADP, ATP, AMP, and they keep going back and forth in time. There is a genome rearrangement sorting by reversals, and it's you can see here it's like the billiard table, how the billiards are scattered and then they are collected again. This is how the genome creates reversals. This is reversal via resonance. You see, this is an incomplete circle, and via resonance, you will go back in time and form a complete circle. What is resonance? Resonance is a way of describing bonding in certain molecules or ions to combine in a sort of what we call a resonance hybrid. And you can see here, this is a formula that creates a hybrid between two different elements. Basically, it's like when a boat crosses through the ocean, these waves end up in bigger waves and bigger waves because waves add up onto each other through resonance to make bigger waves. They amplify. And resonance transfer is something you can see under the microscope with proteins, and proteins use energy transfer in order to communicate, but also to repair themselves. And this is an experimental thing of Fred that you have a donor and an acceptor, and then this is the resonance that creates between the two of them. It's a hybrid. So the two of them are combined. It's like man and woman create a child, basically. It's this type of thing. And again, you have a donor and an energy transfer. And this is the microscope that you need to utilize to see these things. You can see it's a very complex technology. Now, the origins of skin disorders starts with the structure of the DNA. And thankfully, we have DNA repair that can happen through direct reversal, or it can happen with the replacement of the damaged nucleotide. And the lower the element, the more simpler the element, the easier the repair. So here, chromatin is rearranging nucleotides, and here an enzyme is instructed to basically repair a broken strand of the DNA. You're going to see this in a movie in a second. So this is how a DNA mutation will be created. The DNA polymerase copies the DNA, but as you copy it, A and T and G and C have to be matched together. If you have an A with a G that's a mismatch, that can actually cause cancer. That simple thing can end up with cancer. So immune system proteins will scan the DNA and will ask other proteins to come in their assistance to combine themselves and remove the damaged DNA part. Once the damaged DNA part has been removed, then the DNA polymerase enzyme will come and will sort of put those strands together, repair it, glue it together in a way, and then you have an undamaged DNA. And this is how signaling, basically a signaling correction is how a mismatch of DNA, and then you basically add the part that is missing. This is how it's translated in terms of letters. This is how it looks in terms of a genome. And again, a nonsense mutation is very, very simple. You see just one letter changes, and the whole thing can can fall apart. You can have aging, disease, and very serious disease. What's very important to understand, however, is that the same signal can enhance and suppress cancer, for example. So the same signal, depending on its context, the context is extremely important in this molecular biology. And what resonance signals do, basically, you see this is a broken sequence, and it basically fills it in to make it meaningful, to make the words meaningful. Of course, this one you can probably understand it in your mind because you have a gestalt mind, but the body at the molecular level, one letter difference, cancer can be the result of it. So our research is centered about utilizing signaling technology to repair faded or degenerated cellular signals. And you do it through amplification, or you can do it through basically putting in the signals, repairing the signals by adding the missing signals. And there's 5 basic mechanisms of signaling. You need the blueprint signal to form. Then you need ultra-low proprietary energies at which the ion channels of the cells open up. It's a mathematical formula. I'll give you the formula later on. The signals have to be emitted in discrete times, and basically multiple signals have to be emitted simultaneously to create the context. So it's a very, very complex technology. And of course, there's a mega antioxidant effect because of the electrons. And as you can see here, nothing is really accurate, not at this point, not in this state of the art. We're looking at clusters of signals. We cannot see things, in greater accuracy. So, and then waveform composition happens by adding different waveforms together from 6 to 10,000 waveforms, and each one of them represents a signal. Now, this book came out in 2008, and it showed that there's a mathematical formula that proves that at certain energies below thermal noise, the exact opposite of lasers, of high energy, of ultrasound, it's extremely very, very small energy. At those energies, for some reason, the ion channels of the cells open up the way you open the garage door with the remote control. And this is what thermal noise is. Basically, you see the thermal noise, you can see like 0.00 volt, microvolts. It's tiny, it's imperceptible. Now, the other component is the time dimension. Our cells have a circadian clock. Like you have a circadian clock, like you travel and then you cannot sleep because your circadian clock is all out of whack. It's the same thing, our cells have a circadian clock, and the circadian clock of our cells is located in the suprachiasmatic nucleus in the brain. Now, this research was established in Nobel Prize 2017, and these investigators, for very long research, they basically showed that molecular mechanisms control the circadian rhythm and how that happens. And cells are governed by their biological clocks. So you have to deliver the signals in a time sequence that matches that component. Again, another dimension of complexity. But it's quite important, however, because recent studies have already linked skin circadian clock to psoriasis. And psoriasis inflammation is due to disruption of signals delivered at the time schedule. This is a signaling technology study of psoriasis. This is another clinical study on psoriasis again. And how do we identify skin disorders? We can use forward genetics, or now more recently reverse genetics, basically by looking from the problem, analyzing the problem all the way to its root. And this is basically how the bullosa simplex, epidermolysis, the EBS, was discovered through this reverse genetics. And also, we need to look at viruses and how viruses affect DNA. And we have 3 different aspects: that the cytocidal, the persistent, and the transforming. And you have morphological, you have biochemical and physiological changes, and they can inhibit the DNA, they can create inflammation, they can inactivate tumor suppression proteins, and you have different levels of viruses and how they affect the body and how you can basically combat them. For example, the zoster virus is this regulates the human foreskin fibroblasts. This is a signaling clinical case on herpes zoster. This is after 2 treatments. And that's again from the side. You can see all this damage. Actually, you cannot even see with this woman, if you saw it in real life, the damage seemed even more intense than what looks in the picture, and yet it was quite easily repaired. So can we actually reverse aging? Can we reverse illness? Can we unboil an egg? Because what's an egg? An egg is basically proteins that have been denatured. Can we unboil an egg? You're going to say no. And actually now the science is telling us that by refolding proteins, we can actually unboil the egg. And unboiling an egg can lead to better cancer treatments. There's a lot of papers in Nobel created right now. This is how a normal translation can go, and this is a nonsense translation can go, and you can see this is a protein that is not a fully folded protein. And what happens with misfolded proteins? Either autophagy, which is like a vacuum cleaner in our system, it just cleans up the system, or you can refold it, of course. Otherwise, all of them, they just aggregate together and they create skin disorders and aging. Radio frequency, anything over 10 megahertz and to 300 megahertz and over leads to protein denaturation, which is unfolding of these proteins. And in a cell, if you denature a protein, basically it cannot function. It's like a boiled egg, that's it. I mean, it can never turn into a chicken. But the problem with the natural proteins is that they can result in cancer, Alzheimer's, and diabetes. And here you see how a protein that has been denatured can actually be renatured by folding. And there is many techniques of folding proteins. There is the pulsable renaturation process and using chromatographic procedures. There's other procedures that they are slowing down the aggregation, and they sort of, because you need to kind of untangle, it's like tangled jewelry. So you have to untangle them before you're able to refold them. And again, you need to understand that a folded protein is like a gestalt. It's more than the sum of its parts, which makes refolding extremely complex and difficult. It's not like you can just fold it back. Because again, it has to be folded in its exact way of being refolded in order to kind of be functional again. And it has different structures, like the primary, secondary, tertiary, and obviously the higher level of structure, it's the more the complexity and the more difficult it is to refold it. Different techniques, surface plasma resonance, cyclic voltammetry, chronoamperometry, all kinds of different techniques for refolding proteins, and we have a way of actually measuring whether these proteins are folded or not, and it's called the second virial coefficient, the SVC, it's easier to pronounce, and a negative SVC means that protein-to-protein interactions are negative, and therefore their proteins are aggregated, and they're not really properly refolded. That is the bottom line. And a positive CVC shows you that protein-to-protein interactions are much better, and that the aggregation basically has decreased, and therefore the refolding can be successful. This is, we continue international research. Some of it is clinical, most of it is clinical. Our lab research is all proprietary, and this is some of the doctors that are involved in this research. And thank you very much.
Thank you very much, Dr. Sofra. Would you like to take questions
Yeah.
now?
Any
Do you
questions?
have any questions for Dr. Sofra?
I know it's a difficult kind of subject that's a little Yes.
Yeah. How can we actually prevent this from happening? Because when it happens, we know how we can't really tackle it. However, how we can prevent this from happening?
Okay, so heat shock, heat, all kinds of damage, free radicals, all of those things, pollution, all of those things will affect both the immunity, and once they affect immunity, they're going to affect they're going to the natural proteins in DNA. There is, of course, some genetic components, but primarily, if you walk out in London, some of your proteins will be denatured. Heat treatments, like for example radio frequency, it might be good to have another technology after radio frequency that will refold the proteins. So whatever benefits you get from radio frequency can sort of maintain the health status by doing that. So it's not just one thing that can do it, it's many different foods. Your foods, toxicity in your foods, that's why we age. I mean, otherwise, inflammation with the natural proteins, viruses, all kinds of things.
Will the antioxidant diet, for example,
Yes.
whether actually in a supplement or whether actually through natural diet,
Of course.
slow that down?
Absolutely,
Yeah.
it will. Yes, antioxidant will slow it. Absolutely.
What other technology that will actually, help the radio frequency?
Well, you have signaling technology that you can basically utilize to help proteins refold by giving the signals back to the proteins through the resonance process that I was describing. It's a complex technology, it's very new, and it's still getting developed.
Will injectable platelet-rich fibrine help the radio frequency, for example, the consequence from radio frequency to
Everything affects the body, so either in a good way or in a bad way. So everything will affect the body. Is just the more you reinforce your immune system, the more you cleanse the body, the more you relieve it from free radicals. Of course, that's inflammation is great for the young, is deleterious for the old. We need to avoid inflammation. Inflammation is aging. Yes.
So are you suggesting with the menu for our radio frequency skin tightening treatments that we perform, that we give them a diet of probiotics?
Absolutely, yes.
And whole food, plant-based diet?
Yeah. That's a very good idea, yes. Yes. Absolutely.
Thank you very much, Dr.
Oh,
Sofra.
this lady had a question too.
It kind of answered it. Hopefully it's a really quick question, but for me this is all very technical.
Yeah.
What does a signalling treatment look like? How did you get the woman's things gone, the
Yeah.
disaster gone?
Okay,
What does it look like?
it's a
Is it
transcutaneous treatment. It's a device that you can use basically, and it's a transcutaneous treatment because of those energies. The ion channels of the cells open up.
Okay.
Then you can insert signals. You can also insert stem cell extracts or any other substance. Because they open up. It's like a garage door. Car can go in now. So it's. I can give you this lecture if you give me your WhatsApp or something. I can give it to you so you can see that you're on time.
Thank you.
You're welcome.
Thank you very much. We're going to move now to Dr. Jolie.